Good morning, everybody. I am very excited to be speaking today here at the NeuroAI Workshop. So I'm Lisa Schmors. I'm from the Hertie Institute for AI and Brain Health and the University of Tübingen in Germany. And today I will be speaking about contrastive learning for multi-trial time series data in neuroscience. I actually wanted to start this talk by drawing your attention to a very recently published review. I thought it might also be a very good start for people who might not be super familiar with the topic in the audience, but might be very interested in. So the review is called The Emergence of NeuroAI, Bridging Neuroscience and Artificial Intelligence. And the authors, they start by saying that the most immediate and transformative promise of NeuroAI lies in tackling neuroscience's big data challenge. And indeed, if we think about neuroscience is creating terabytes of data every day, starting from single cells recorded to connectomics, where you are interested in the connectivity between neurons, behavioral neuroscience studies, or neurology. And I think today we have a very big potential of generating insights to an unprecedented scale into how the brain works. However, it can also be sometimes very confusing because often data is high dimensional and we need to be very careful with the approaches that we are using to make sense of this high dimensional big data sets. And the question really is, where can AI come in and help us understand this data? And today I want to speak about one specific approach to make sense of high dimensional data, which is using visualization tools to create lower dimensional spaces, to be visualizing high dimensional data in low dimensional spaces. Okay, so think about a system where you have many different neurons. They are all different, which you can see here by the shape and the color. And the question now is, how do they relate to each other? Where are they different? And where do they share similarities? So in neuroscience experiments, we often have a bunch of cells, thousands of cells, measured, and we measure different features. Can be the size of the cell, can be some transcriptomic information, so the genetic information, protein expressions, but it can also be activity over time. So in this case, the features would be the time point, and then the value would be the activity measured over time. So we often have data matrices where the cells are the rows and the features are the columns. And we have many features, maybe thousands of features. And now the goal would be to create a low-dimensional embedding or a low-dimensional representation where every dot now is a neuron. And you can see that we can identify different clusters. We can see how the clusters relate to each other. We can also identify outliers. So there's this like little orange cluster at the top right, that might be an outlier. And for humans, it's much easier to interpret spatial relationships in this two-dimensional representation here compared to these like numerical relationships that are in the feature matrix that are very difficult for humans to interpret. So it becomes clear that these low-dimensional representations can be very powerful. And in this talk, I want to explore today if we can actually exploit the specific data structure or the specific structure in general of neuroscience experiments, if we can exploit the structure to create low-dimensional visualizations specific for neuroscience data. And for that, I would first like to introduce to you the data set that I would be working with. And that data set was recorded in collaboration with Katrin Franke, Jack Raymer, and Andreas Tulliers at the University of Stanford and the Baylor College of Medicine. Okay, so the brain area that we are in is the superior colliculus, and we have retinal neurons, visual neurons that project to the superior colliculus. And my collaborators, they recorded the activity of neurons using calcium imaging. And you can see two example neurons responses in the middle in this black and white heat map. And at the top, you can see a little squiggle, which is the visual stimulus. So that stimulus is a kind of barcode stimulus that has been used throughout the years to identify the cell types of visual neurons. It's kind of a flicker from white to black. You have some contrast sweeps and some frequency sweeps. And for these two neurons, you can see that in the first 4 seconds of the responses and of the stimulus, they already are very different. And these different responses we can now use to identify their functional cell type. Very important for this data set was also that we repeated the stimulus 15 times in this case. So we don't only have like a single trial or a single repeat response of this neuron, but 15 responses. And that is a very common structure in neuroscientific experiments where you do one thing many times, measure the same neuron multiple times to get a better idea of what the neurons are actually doing. In total, I had over 70,000 neurons in this data set, and on the right you can see my attempt to sort neurons and to get an idea of what kind of functional responses I have in this data set. But here you can already see this is high-dimensional data. It's very hard for us to even figure out how many cell types or how many clusters might there be present in this data set. So I developed a time series representation analysis through contrastive embeddings. And I will be going more into detail of what contrastive embeddings are, but I called my approach short trace. And here you can see a 3-dimensional representation of this data set. Again, every dot is a neuron, and they are colored by the identified cell type or the identified cell cluster. And you can see them sorted around, and you can see their response over time that belongs to the cell type. All right, but let's start from the beginning. So I told you already we have this calcium neuroresponse for every neuron. Let's start with one neuron. And then in contrastive learning, it was first developed for images, and the idea is to contrast samples to find where they share commonalities and where they are different. And on the top right, you can see the loss function. And in this approach, we try to maximize the similarity between points that are very similar. And so we try to put them close together in embedding space, and we try to put the samples that are very different far apart. So if you think of the picture of a dog on the top right, we generate these embeddings by creating 2 instances of the sample. For this image of a dog, it could be we create a crop out and we rotate the image. And this is our positive pair that we try to minimize the distance between. For our neuron now, that could be 2 instances of its response. I told you that we recorded the neuron 15 times. So what we do to create this positive pair was to select randomly 7 trials and compute the average, and select 7 other trials and compute the average. And that would be 2 representations, 2 samples for the same neuron. All right, so that's our positive pair. We also need a negative pair where we maximize the distance between those 2 samples in our embedding. And for the picture of the dog, that could be a picture of a cat, maybe in gray. So we want pictures of different animals to be far apart. In our case, for the neurons, we just take a random other neuron in that batch and say those 2 neurons should be far apart. Okay. And then in contrastive learning, you feed your samples, could be images. In our case, it's time series data. You feed it through a DNN. And then the last layer of your deep neural network defines the number of output dimensions. So in the animation that I showed you, I had 3 neurons in my output layer. Could also be 2 or 10, whatever you are interested in. All right. And then we have our low-dimensional embedding, and you can see that we minimize the distance between the instances of the same neuron. So we would hope that another neuron that responds very similarly would be very close in the embedding, and we maximize the distance to a neuron that has a very different response. Okay, let's recap. So I presented to you a contrastive learning framework for 2D visualizations of time series data. I presented you a novel data augmentation techniques using the means across a subset of trials. And that allows to automatically use the statistical structure. Instead of coming with data augmentations that might be very artificial, we can use the structure that is already there, and even better, for every neuron. So every neuron also has a very different statistical structure, and in the way of using the subsets of trials, we can really use that structure. And we find distinct clusters in the embedding that corresponds to cell types. I briefly want to show you results for a different data set. So this data set is public. It's a bunch of visual neurons also responding to light stimulus, recorded and published by the the Allen Brain Institute. And this data is a little bit different. While we had calcium imaging data, this data set is a spiking data set where the resolution goes down to the single neuron action potentials. And especially in this setting, our method was very beneficial because traditional data augmentations for time series data, they, for example, include Gaussian noise. You can change the amplitude, you can jitter the temporal structure of the signal to create the positive pairs, but that is very artificial. And spiking neural data, action potentials, they follow a Poisson distribution. And by our idea of using the subset across the average across a subset of trials, we can really make use of that statistical structure. Okay, that's just the beginning, because while working on this project, I actually realized that there is so much potential for this approach to also be used in different areas, from climate data to financial market data to sports analytics. So currently I'm looking for different data sets where we can maybe reshape this data into a multi-trial structure. So for climate data, for example, you can think of different repetitions or different trials being days or maybe tidal cycles or years. For sports analytics, this could be games. Maybe you are interested in the dynamics of players interacting with each other. And for medical data, I could think of a data set where the same patient is recorded multiple times in a task, or maybe also like heart rate data over days. All right, and then I want to finish my talk by also mentioning a future project that I'm very, very excited about. So I will be collaborating with Joseph Raimondo at the University of Cape Town, and one of his postdoc, Thais, he recorded a data set over the past 8 years, I think, from epileptic patients. And that is a huge and rare opportunity to actually study the human brain. And I'm particularly interested in biophysical models of these human cortex neurons. Yeah, so I think Alana later will also talk more about Jaxley. That has been a tool developed by our lab. And the nice thing about that is that with Jaxley, we are able to perform backpropagation through models that capture the biophysical dynamics. So far, that has only been possible through simulation-based inference, for example, or evolutionary algorithms to fit these biophysical models to neural data. And now Jaxley allows us to do that with error backpropagation and optimization, which is, I think, a game changer for the field. All right. So I also wanted to draw your attention to the Imbizo, which is a 3-week computational neuroscience summer school in Cape Town. You can scan the barcode at the bottom to get more information. And they cover broad topics from molecules, behavior, decision-making, and also neuro-inspired AI. Yeah, and if you have any questions or the application process, you can also come to me. All right, and with that, I would really like to thank you for your attention. I would like to thank the whole lab, the whole Behrens lab, so the lab of Philip Behrens, and specifically the people who are working with me on the TRACE project, Dominic, Nick, and Sebastian. Yeah, and thank you for your attention.
So we're taking questions and answers now. Any questions? Just
Thank you for your presentation. That was very insightful. I just have one question. Maybe I might have missed it. So you were studying this time series, but I didn't see if when you take this into the deep neural network, you actually learn the time dimensionality itself. Or is it you don't learn it because you compress that using the mean?
Yeah.
Okay.
That's exactly the point.
Okay.
So the input dimensions would be the number of time points that you measure in your time series. And of course, that is depending on the temporal resolution. You can always downsample a signal. But we had, I think, our first dimension was around 600 bits or like time points of our time series data. And then we would compress that information. We would treat every time point as a feature and compress that information into 2 or 3 dimensions. Yeah.
Thank you.
Hello, nice presentation. I want to ask about usually time series data have like a standard frequency, like it doesn't change. So if I'm wrong, I want to confirm the frequency of the data is 0.2 milliseconds.
Well, that really depends on the data set. I think in our case, it was 0.03 seconds. So, yeah. So, but that really depends on the binning that you decide to do. And I mean, you have a lower bound by, for example, neurons that have a very high frequency response to these amplitude sweeps and contrast sweeps in the stimulus. So there is a lower bound. But if you have a step response, for example, like, yeah, you could go even lower.
Okay. May I finish? Yeah, thank you. So you calculate the mean on 10 samples, and the 10 samples, like the frequency is the same.
Yes.
Is there a chance that you don't get one sample, like for example 0.2, and then you don't get a 0.2, you get a 0.4, for example, like you missed one data? Yeah. Do you like it's like a missing value, you know? Yeah. Do you fix it and then you calculate the mean, or you just skip it and to the next one?
Yeah. So in our case, that didn't happen because perfect. We have this microscope that is recording the activity of the neurons in a very determined temporal frequency. But actually, yeah, so you can compute the mean even if you have missing values. Of course, you should check. And if you have many missing values, maybe you should exclude that particular neuron or that particular scan. But I think especially for our approach, that is very powerful because you have other trials that have a data point recorded at that moment, right? That gives you an idea of the activity of the neuron. While for other contrastive learning approaches, you would try to add some noise and there is missing value and you don't know.
Yeah, thank you so much.
Thank you, but that's a very good point.
Thank you.